let me begin. I hope all of you, I think all of you guys know me, and those who don't, I will say that I've been at PHRI for 34 years, and I have received much love and support here, and that continues from Rutgers. PHRI is extension of my family, and some of my family members are also joining. In fact, they joined me after I joined PHRI, and sure. Go ahead. So it's really a in a normal year I would have brought cookies for you that Ritu would have made, but this is a special year, and so we're going to do it without cookies. Place a pointer. So I'm going to talk to you about our work on noise in gene expression. This is one of the main things our lab does for many years. And in particular, I will discuss these 3 papers that I've listed here. First published back in 2006. Describes the first description of noise and gene expression in eukaryotes by Arjun Raj. And the second describes what kind of consequences cells have to face as a consequence of the noise in gene expression. And the third is a paper just published in PNAS by Latasha Fraser that talks about the mechanism of noise generation in eukaryotic cells. So this work began in our lab back in 2004 when Arjun Raj came into the lab as a graduate student. And Arjun was a mathematics major from Courant Institute in NYU, and his advisor, Charlie Peskin, sent him to our lab to do some project that will have possibilities of exciting math. And at that time, he just didn't know how to pipe it. And today, he is a star in biology. In fact, a experimentalist, the best experimentalist you can have today. And so I'll start with this paper that we had in front of us back in 2004. And these folks in Alovitz and the other folks at Rockefeller University, they studied how gene expression varies from cell to cell in E. coli. And a priori, you would expect that all cells are identical in terms of expression of particular genes in E. coli. They have one copy of the gene, and they have few copies of the transcription factor that would turn them on. And they did a very simple experiment. They had a copy of CFP under some promoter and a copy of YFP under the same promoter. And these 2 copies of the gene were inserted in the genome of E. coli cell. And then they simply ask how much of CFP and YFP each cell is making by imaging them in these 2 colors. And this very evacuative image shows that cells are so different from each other. Even daughter cells differ in terms of how much of each protein they make. And you expect that if laws of mass action were operating here in a classical sense, each cell would make about equal amount of CFP and YFP. And what you see is a complete departure from that. And the reason for this was very simple. These folks described that the number of copies of transcription factors in E. coli is so small, it varies from 2 to 5 copies or sometimes none, that at any given moment, either gene is on or it's off. So,
Sanjay, can I ask a very naive question here?
Yeah, of course, yeah.
So in a synchronous culture, would you still see the same expression profile?
Yeah, in fact, these guys went to great lengths to synchronize the culture, to mix it nicely, and so on. In fact, these two genes were placed equal distance from the origin of replication in E. coli chromosome. And this paper has become a cult classic in biology, and it started a huge field. And there's, in fact, no doubt about this phenomena at all anymore. So when Arjun came to the lab, we said we're going to do this, study this phenomena in eukaryotic cells. And in eukaryotic cells, the number of transcription factors is much larger, varies from 10 to 50,000 copies per cell. And so we had developed a pretty simple and nice way to look at RNA at single molecule resolution at that time. And the idea was that we would look at RNA rather than the protein. We look at protein as well, but we focus on the RNA because RNA is the most immediate product of the gene activity. And the way we visualize single molecules of RNA was to engineer these genes coding for fluorescent proteins so that in their 3' untranslated region, there would be tandem repeats of a 50 nucleotide sequence, about 32 repeats. And we would have probes that contain 5 fluorescent dyes. So using these probes, we could deposit about 150 molecules of fluorescent dyes on each RNA and see each molecule of the RNA as a fluorescent spot in microscope. And these genes had additional features that allowed us to control them. So one feature was that they were doxycycline controlled, meaning when you remove doxycycline, they would express. When you add doxycycline, they would turn off. And we would image these RNAs in Chinese hamsters' ovary cells. And these cells are cultured on a glass cover slip and fixed by formaldehyde. Their membrane is permeabilized by adding alcohol that extracts the lipids. And basically you have a sponge-like matrix that traps the RNAs where they were at the time of fixing and allows probes to go in and out. You add the probes, you remove the excess of the probes, and you image next day. And you see these spots in the 3 dimension of the cell, which you can count in the 3 dimension. And Arjun wrote computer programs to analyze these images that precisely identify each molecule and count them in each cell. And we went to great lengths to show that these are all the molecules of that RNA that are present in those cells, and we don't miss any. I'm not going to go into those experiments, but what he did was he counted these RNA molecules per cell, sort of developed a census of this cell population. And I'm going to come back to that image. And this census was presented in these histograms. And so to show you this histogram a little bit closely, it has this bins of number of RNA molecules. For example, 55 cells has 0 to 10 molecules, about 9 cells at 10 to 20 molecules, and so on. So these histograms are very odd. They are more like wealth distribution in human society. Most people have very little money, and few people have a lot. And the mean of this population is decided by high performers, these few cells that are making few hundred molecules, not by the average person. So if the cellular society was like Marxist society, it would follow a Poisson distribution, meaning almost all cells would have about equal number of molecules. And you can see it departs massively from that Poisson distribution. And how much it departs is described by this statistical factor called Fano factor, which simply is a standard deviation squared and divided by mean. So if this is the standard deviation of this population, you divide that by this mean, and you'll get Fano factor. And for Poisson distribution, it is by definition 1. And you can sort of see our cell population had a Fano factor of 200. It departs greatly. And by comparison, E. coli cells had a Fano factor of 2 or 3 or 5. So these mammalian cells depart great deal from the average behavior. And so at that point, we thought, okay, what is the origin of this phenomenon? And we suspected that it comes not from global factors in the cell, like cells could be in a wrong stage of cell cycle where the RNAs are not made, and that could produce these non-performing cells. But the cells divide rather slowly. Their doubling time is about 18 hours, and you can see that that can't account for this. So Arjun did a very clever experiment. He said, okay, I'm going to take 2 genes that code for a fluorescent protein and have 2 different kinds of tandem repeats in their UTR. One we could probe with green probes, and other we could probe with red probes. However, he took these 2 genes and integrated them in 2 different ways in the cells. In one case, the 2 genes went to 2 different loci in the nucleus. In another case, they went at same location in tandem, tandemly. They were next to each other, and these 2 cell lines behaved very differently. The one where you had 2 different, where the genes were in different locus, they showed either expression of green or red, and only occasionally both. And if you just simply look in the red channel, and you would see the cell is in the wrong stage of cell cycle, but you can see that same gene under the same promoter is being made at other location. However, and this is true statistically for many cells, they either make a lot of red or little or a lot of green, and very few cells make both. However, when we examine the cells where the 2 genes were located next to each other, they always made both the genes together, and that was true statistically, and because now you can see most cells lie on this diagonal. If they make a lot of red, they also make a lot of green. So this told us that there is stochasticity in the gene expression, but it lies at the chromatin. And I'm going to come back at it in a detailed manner towards the end of the seminar. But we developed a mathematical model that basically describes the status in one cell for that gene. And the gene undergoes, even after you turn it on, meaning remove doxycycline, it doesn't stay on. It goes on for a while, goes off, goes on again. And during the on period, it makes a lot of RNA, a burst of RNA. And burst can produce maybe 50 to 150 molecules. And once you make the burst, the gene goes off and the RNA simply decays. And over time, a particular cell experiences this burst and decay. And depending upon when you had fixed the cell, you would see, okay, this cell is experiencing a burst, while these cells had experienced this burst of RNA a while ago, and the RNA had decayed. So in fact, you can sort of see this telltale sign of the burst happening at the gene locus with this big cluster of RNA still tethered at the gene locus. I'm going to come back to this later, this phenomenon. So basically what we are seeing is that genes experience periods of on and off randomly. And one more important thing to consider is that how do the cells vary in terms of number of copies of RNA and the amount of proteins that is produced by this RNA. And you would see that we can look at the same set of cells both for the protein and for RNA, and you will see that these two cells here, they make huge amount of RNA and huge amount of protein, but consider this pair of cells, this had very little RNA and this had a lot, and yet they have about similar amount of protein. So what happens is that RNAs have short half-life, so their decay is fast. So this cell made a lot of RNA and then decayed, but protein have much longer half-lives. They are resistant to these variations and they buffer the variations. So this is important principle that has come, that longer the half-life of the marker protein, less resistant it would be to the variation. So I showed that even though proteins are buffered in terms of variation, there is a great deal of variation. So how would the cells deal with this? Imagine that you have a pathway where you have a transcription factor A and it's variable, and it induces expression of a gene B, which induces the expression of C, and so on. There could be such a pathway. And if you had a cell 1 that made 10 copies of A and its neighbor made 20 copies of A, you can just imagine that by the time you make D, you will have made 100 copies of D and 5,000 copies of D in cell 2. So this noise, this variation will amplify in a simple gene expression pathway. And does it, and how do the cells deal with that? Because their phenotypes will diverge, and rather than seeing almost identical cells in a tissue, you would see greatly different cells. So cells have to deal with this phenomenon. And so we examined this, and I'm going to take a few minutes to explain, to describe this project that we did with Khyati Shah when she was a graduate student in the lab. And what we did was we said, okay, we're going to look at, in HeLa cells, some genes that are induced early when you add serum to serum-starved set of HeLa cells. And these, so when you do this, you starve HeLa cells for serum, they don't divide anymore. They come to stand still in their cell cycle. And when you add serum, they begin to divide. And that program of division, start of division, begins with synthesis of these transcription factors, these early response genes that are made quickly and then decay quickly. And their products go back into the nucleus, turn on the secondary genes that are respond a little later. And people, early microarray work was done with this system, and we picked up a set of a pair of early response genes called C-Force and C-Jun. And these are classical transcription factors. They're sister proteins. They bind to each other, migrate into the nucleus, and turn on maybe 50-some proteins and turn themselves off. That's key, an important consideration. So these guys, these early guys, they engineer their own production on, they engineer their own gene to turn off. So basically they go back and serve as a repressor for themselves. And that leads to loss of their amount of RNA. And so we said, okay, we're going to study the expression of C-force and C-jun, and then we look at the expression of a downstream pair of messenger RNAs that they turn on. And so we are going to look at them by SM fish and both the starter and the end product. And so to do this, you remember the first set of genes we visualize by engineering the gene with tandem repeats and probing them with those multi-label probes. That is not feasible for natural genes. And so we decided we're going to have a new system that will be able to detect RNA for natural unmodified proteins. And basically this system works the same way as the previous one, except here we make unique probes spanning the entire body of the RNA, about 50 probes that will bind to the same RNA and deposit many fluorescent dyes. And this system works out very, very well. It's very popular. We licensed this to a biosearch technology, and many, many labs around the world use it now. And you can see the kind of results you get. Here is, we are probing HeLa cells soon after induction, 30 minutes after induction with serum. And you can see, again, this cluster of RNAs at the gene locus for C-force gene, and these single molecules distributed in the cells. In the same cells with probes labeled with a different dye, you can now look at the C-jun, and you can, using a computer program, and identify the spots and explicitly count them. And you can also see where the gene locus is. So right away, you see this image, and you will see that the C-force and C-jun proteins that are sister proteins that are going to form a heterodimer are expressed in very diverse and uncorrelated manner. You can see statistically that in this graph, I'm showing you the C-jun RNA molecules in a given cell. So each dot comes from one cell. So this cell made about 175 molecules of C-jun and 300 molecules of C-force. And so there is no correlation. After all, these two are separate genes. So these two are expressed randomly, like their phenofactors are very high, like I'm saying, 171 or the 30. And however, and we want to correlate this variation with the variation in the downstream genes. But what we face a problem that these RNAs disappear soon after their synthesis. So by the time the downstream genes are made, these RNAs are not there anymore. So the information is not there at the time of the downstream RNA synthesis. So we had to come up with a way to look at the heterodimers in a quantitative manner in single cells. And what we did was we developed this technique called proximity ligation assay. Rather than develop, we adopted this. It was actually developed by Ulf Lentergren in Sweden. And the way this technique works is that you want to detect only the heterodimers and not the free proteins. And the way you do this is you have two antibodies raised in two different animals for each protein. And when they bind to this heterodimer, it brings these oligos that are attached to the antibodies close to each other so much that they can mediate circularization of these two additional oligos that you add. And if you add ligase and then a DNA polymerase, you create a tandemly repeated copy of that circle. And in that tandemly repeated copy of the circle, you can probe that with a probe and detect this heterodimer by fluorescence, and you get results like this. So this is C-force C-jun heterodimers 6 hours after induction. And each spot, you would appreciate that it is present only in the nucleus, and it is what is surprising is that even though the RNA was highly variable, the protein is, or these heterodimers are not. In fact, if you do statistics on it, their Fano factors are pretty low, and their distribution is close to the mean. So even though the proteins were made in highly stochastic manner, the heterodimers in the nucleus are present in sort of reliable and low variation manner. And so the variation in the parent molecules of RNA is sort of decreased, and the complexes that are going to turn on the gene is present in a reliable manner in almost all nuclei, about 50 copies or so. Okay, so question is, when you look at the downstream genes, what do you see? I'm going to move this away. Sorry. So here I'm showing you the two downstream genes, COX-2 and collagenase 1, now along with the heterodimers. And you can sort of see every cell has, every nucleus has heterodimers present, and only this cell makes collagenase 1, and only these three cells make COX-2. And so even though the system had regularized the expression of transcription factors, the heterogeneity in the expression of the downstream genes come back. And if you look at it statistically, you can sort of see this gene, collagenase 1, has very high Fano factor of 120, meaning most cells are silent, few cells make the RNA and decide the average. And same thing is true with COX-2. And so the system needs the heterodimers because in absence of it, you don't make these genes, but it doesn't matter how much of that exists. So I'm going to summarize this. Basically, there is a sort of a bottleneck of noise in gene expression. The C4 and CJUN, the transcription factors, are expressed in a noisy manner. Their heterodimers are sort of, noise in them is low, but the noise comes back and in the downstream genes. And it's simply a consequence of transcription being intrinsically noisy. And however, in that data lies this idea that we don't see amplification of noise that I was worried about in the beginning. You see presence of very, very noisy behavior in both downstream and upstream genes, but no evidence of amplification of the noise. And this is very good for the cell because if there was amplification, the cells would go out of sync. And I should say other labs have found and explored other pathways to minimize the impact of this noise. And in fact, there exist sometimes duplicate pathways that lead to the same product to simply minimize the noise in the impact of the noise. I'm going to leave this impact of the noise issue here and now dive into the issue of where does this noise come from and what is the molecular basis of this noise. And the Arjun's work back in 2006 had suggested that noise sort of resides in chromatin, meaning if the two genes are close to each other, their noise is, they sort of fire together. If they're far away, they are uncorrelated. So there's something at the chromatin level that controls it. And so this is an exciting, new, vibrant field now, and many, many labs are working on different hypotheses to explain this. One of them is that there are multiple transcription factors that need to assemble at the gene locus to start producing RNA. And that assembly of complex machinery becomes stochastic because there are so many elements that must come together at the gene locus. A second idea is that in the nucleus, people have seen that there are about 100 places where there is massive transcription going on. And these are transcription factories, and the gene has to be close to them. And an arrival of the gene to these factories becomes stochastic. Another idea is that for a productive and high capacity transcription, a distant DNA element called enhancer has to interact with the promoter, and that interaction is stochastic. And there's this idea that many polymerase molecules are flowing through the gene, one behind other. It's like buses going on a avenue in New York, where if you're waiting for a bus, you wait for a long time, and then many buses come together. And basically, towards the end of the gene, all these polymerases come out together. So they it's like traffic backing up. That idea is still around. We work on this idea called random chromatin accessibility hypothesis, and I'm going to spend the rest of the time on this idea. And this last facilitated reinitiation is part of our hypothesis.
Sanju, should we hold the question until the end, or can we ask question now?
You can ask a quick one, not to distract me too much.
Okay, just the quick one is that for the fossil genes, when you do the Western blot, do they correspond to the mRNA?
Yeah. So please don't worry about those kind of things. The C4 syncytium.
I'm just curious that with the heterodimers, that's only represent a subset of the protein, and actually the total protein actually sort of similar to the mRNA profile.
Okay, the total protein, we actually explored total protein by simple immunofluorescence as well. And so in terms of there was high variation. I'm going to go back here. Let me go back for a second. So in terms of the variation in C4 syncytium, they reflect the variation in the RNA. So we measured the variation in the individual proteins as well, but key idea is that this heterodimer in the nucleus is much less variant than the parent proteins, which is less variant than the RNA.
Okay, thank you.
So half-life of this RNA is only 6 minutes. Half-life of the protein is about 40 minutes or so, and half-life of these heterodimers could be days. And I think that's the key driver of this variation. Mm-hmm. Okay, I'm going to
ask a question.
Let's save them, because I think this is the real meat of my seminar, and let me not get distracted on the old work. And of course, we will answer all the questions. Just remember them. And so the idea is that the nucleus has 10,000 or more copies of the transcription factor, 2 copies of the gene, but the gene is not accessible. It's sort of hidden, sequestered in the chromatin. And some kind of breathing in the chromatin allows the transcription factor in, and that breathing is what gives rise to the noise. And that's what is our hypothesis, and I'm going to explore that experimentally. And to do this, what we are going to do is we're going to just look at the COX-2 mRNA. I've just introduced this earlier, and this is expression in a field of HeLa cells 4 hours after serum induction. You can see is highly variable. And so what we did is this is the work of Latasha Fraser, who finished a PhD about a year and a half ago and is currently in NYU. And what she did was she said, we're going to increase the accessibility of promoter at this gene, okay? And people had done some investigation of increasing the accessibility of genes in general by adding a HDAC inhibitor to the cells. And HDAC, are condensing, they promote condensation of chromatin. And these results were, this was done about 5-7 years ago. And as you can imagine, HDAC inhibition pleiotropically changes the structure of all genes. So we said we're going to change only one gene at a time. We chose this gene COX-2, and she took a DNase deficient version of Cas9. I suppose everybody knows this, so I'm not going to give any further introduction. So this DNase deficient Cas9 was fused to a histone acetyltransferase abstracted from a transcription factor called P300, which has a histone acetyltransferase hat domain, which we took out and fused with Cas9. And now if you add a guide RNA in the same cell that takes, that targets the Cas9 to this promoter, you would have deposited a active hat histone acetyltransferase to that gene. So during this engineering, we were conscious that not to compete with transcription factors. So transcription factors for COX-2 have been mapped to this area. So we went little upstream of that, and we tethered this DCAS9 there. And we had actually isolated stable cell lines that express both the guide RNA and DCAS9. So, and we confirmed that they were expressing both of them with little variation, not so much variation. So every cell has these, and every cell would modify this and keep it constitutively open. So the idea is we will ask the cells that constitutively have relaxed chromatin at the gene locus, do they have less noise or more? We did a control remember we studied two genes, COX-2 and Col1, so we made guide RNAs for both. In this experiment, we are just looking at COX-2. So cell line that had guide RNA against COX-2, this is the average gene count, mRNA count. And you can sort of see, irrespective of whether you had unmodified HeLa cells or you had Col1 guide RNA, meaning at this gene locus, which is on chromosome 17, and this gene, which is on chromosome 1, they are not modified. Their average expression is not changed so much. However, when you measure the measure the phenofactor, the noise in gene expression, you see that this cell line where the Cas9 was parked at the COX-2 promoter shows much lower variation than these two, okay? And so this is the key result of this paper that just published in PNAS, and the rest is bells and whistles, which I'm going to talk about. Basically, we are showing that if you and we did other kind of controls, including hat, inactivated hat, and so on. So we showed that this engineering of the gene locus leads to actual acetylation at that gene locus. So if you do chromatin immunoprecipitation, you can show that the status of histone 3 at lysine 27 was acetylated, more acetylated compared to unmodified HeLa. And we also could show that it had more RNA polymerase 2 at that locus. more importantly, Ryan Dicktan in the lab, who had joined about 3 years ago, he carried out genome-wide accessibility analysis of the chromatin. By ATAC-seq. ATAC-seq allows you to explore what sites in the chromatin are accessible. And I'm going to show you in particular only at this gene locus, even though he studied the entire genome of HeLa cells. And you can sort of see that by orange line, this area of the gene becomes more accessible. And these two blue or cyan bars are the locations of the guide RNAs. This is where the hat is tethered, and this is the beginning of the gene. And somewhere here promote the transcription factors will bind. So this gene is open constitutively as a result of this engineering. However, we found that even though, so the whole experiment was, we had engineered had tethering at these two genes. When we examined the variability or the noise in the expression of Col1, we didn't find much change. And that was weird. So we said, okay, maybe it needs more deeper deacetylation at its gene locus. It is more condensed naturally. So we devised a scheme, adopted a scheme. It's called sun-tag strategy, where you don't attach the hat directly to the Cas9, but you attach it indirectly through a tandem repeat. So basically, you fuse a tandemly repeated peptide called GCN4, 10 copies of that to TCAS9, and then have P300 fused to an antibody that binds to that peptide. So you deposit about 10 copies of hat at that location. And when we did this, we found that no Col1 gene was also became accessible, and it displays the same phenomena, that the noise was lower. Sorry for the interruption. So basically, when you attach many more molecules of hat to Col1, that too behaves the same way. So, however, I have a few more minutes, and maybe I'll take only 5 minutes to describe one more cool thing we did with the same system. So, one of the concern is that, okay, it turns out that COX-2 in HeLa cell is present in 4 copies. There are 4 copies of this gene in HeLa cells, and yet it's still so variable. And so, and also another important thing to consider is that this Fano factor as a measure of noise in gene expression is sort of an indirect measure of activity at the gene. It basically measures, it doesn't really see gene turning on and off, it sees the product of the gene expression. So we thought we were going to look at gene directly. And to do this, we image nascent RNA that is being produced at the gene locus. Remember, I showed you this cluster of RNAs that still remains tethered early during transcription burst. So we said we're going to image those bursts by probing for the introns. Now the introns, as you transcribe through a gene, introns are made and they are rapidly degraded. In fact, the pre-RNA doesn't leave the gene locus with the introns. Introns are removed, and then fully processed RNA would leave the gene locus. So if you make probes for the introns, they would be visible only at the gene locus. And here these probes are for green, and you can sort of see cells have sometimes as many as 4 gene loci, sometimes 3, sometimes 2, sometimes 1, sometimes nobody is active. So basically these 4 genes presumably are acting completely autonomously and stochastically. So in this cell, there are 3 copies of gene, and these 3 are on, but the 4th one is off. And this one has only 1 that is on at this moment. So we basically decided we're going to look at how many copies are on at the time of fixing and analyze that. And when we did this, there are 5 possibilities for each cell. It may have 0 copies that are on, 1 copy that's on, 2 copies, and 3 and 4. And the results are right here. You can sort of see unmodified HeLa cells have either 0 or 1, and very few cells have 3 and 4 that are on at a given moment. But this engineered cell line has 3 and 4 that are more common. In fact, the probability of any given gene is much higher in this cell line than these cell lines. So by this measure, by looking at the gene locus directly, you can say that gene turning on probability is stochastic, and that stochasticity is reduced by decreasing the accessibility at the gene locus. There was one more wrinkle, which is you have 4 copies of the gene. What if the 2 copies are present in a heterochromatin area, and the other 2 are in euchromatin area, and your Cas9 system just cannot get to the heterochromatin during its decondensation activity. And people have sort of explored this for CRISPR system. They analyze alleles where 1 allele was in heterochromatin and 1 was in euchromatin, and they ask, can they knock down both alleles with equal probability? And it turns out the results of those work shows that ultimately you can knock down both alleles, but heterochromatin ones are slower to knock down. So we explored this. We actually had a very cool system to look at these 4 sites at the COX-2 gene locus. So the 4 COX-2 genes are almost identical. They are in fact identical. There's no difference. The whole chunk of that chromatin, that genome has been duplicated twice. So we found a SNP, a single nucleotide polymorphism, that is common in this gene in human population. And we said, is that present in HeLa, or the donor of HeLa cells? And so we found there was a SNP that was present in the untranslated region, and we sequenced that, and we are going to use that SNP to distinguish between these 4 copies, okay? And we found that there are 2 variants by this SNP, a G variant and a C variant. How do you distinguish gene loci or mRNAs by SNP? So we had developed a system called AmpFish a couple of years ago, and this allows us to do, and I'll very quickly and briefly describe this. So basically the idea is that you can deposit large number of fluorescent dyes onto an RNA by carrying out amplification in situ. And the way it works is that you will have these 2 probes having these cool hairpins attached to them, bind next to each other. And when they bind next to each other on a particular RNA, they free up the sequence C prime, B prime, which mediates amplification using something called Hcr, which I'll describe. And this Hcr creates a tandem repeat of labeled DNA at that site. So in fact, you can sort of see when you do this for GFP RNA, cells that express GFP has a lot of Hcr signal in the cells that don't. So it's very, very specific. In fact, we converted this. Okay, so I will take a brief moment to explain how Hcr works. This was described in 2004. And the way it works is that you have these 2 mutually complementary hairpins that are labeled with a dye, but even though they're complementary, they don't bind to each other because they're stable. And if you add this initiator will interact, this A prime will bind to the A and create this kind of structure, which resolves into opening of C prime and B prime, which binds to, which is present in hairpin 2. And when it does that, they form a complex, and this goes back and forth until all the hairpins are consumed, and you make a 10 kb long DNA that is still tethered at that location where the initiator was. Okay, so we have developed this AMPFISH using this Hcr, and how we use that to distinguish between S and P is sort of shown here. I hope I can, without confusing you, describe this. So we created cells that had expressed either a wild type GFP or a mutant GFP, and differing only by one nucleotide. Okay, and in this experiment, we have these 4 hairpins, and 3 of them will bind at this location. So this one, this acceptor binds here, that acceptor binds here, and if the RNA was mutant, this mutant hairpin binds and opens that guy, which gives green color. If that one binds, it opens this guy and gives red color. And you can sort of see a complete discrimination between these 2 RNAs differing only by a nucleotide. So we're going to use this exact system to distinguish 2 COX-2 gene loci. And so basically, this is even more elaborate experiment. And what you do here is that we're going to look at the gene locus by FISH against introns. So it's going to light up the gene locus in Texas red. You can sort of see in this particular cell 3 spots, for the active genes. And we want to know which one is expressing a C variant and which one is G variant. And this analysis using the AMPFISH colors, green and red, it shows that this guy, this gene locus is the C variant, and these 2 are the T variants, and the other C variant is not active at this point. So by doing this analysis, you can actually look at hundreds of transcription sites, 500 in this case, and they fall into 2 categories by the ratio of Psi3 and Psi5. And you can see basically, as we predicted, that the number of C and G types, or C and T types of transcription sites are about same. And what we found was, so here, this population comes from unmodified HeLa and COX-2 guide RNA containing HeLa. And when you ask how many of sites in this category were C, they were equal in both unmodified and COX-2, meaning both kind of transcription sites are equally amenable to modification, to decondensation by the CRISPR system, and they're equally likely to express as well. So at this point, I'm going to wrap up and basically discuss this model. The model is that in eukaryotic cells, you make a lot of transcription factors. You have few thousand copies of any given transcription factor, but it cannot see the gene. It doesn't see the gene. Gene becomes visible if some kind of encounter with HATs or some other decondensation enzyme opens it transiently, which allows entry of transcription complex. And once the RNA polymerase complex gets in, it itself recruits chromatin remodeling complex, and it's going to copy through the body of the gene. And during transcription, the entire gene becomes sort of decondensed. And it's going to make many copies of the RNA. And in fact, after RNA polymerase reaches the end of the gene, it's going to find it easier to come back to the same gene than to go to some other gene. And then that is this idea of facilitated reinitiation. And this sort of round back and forth creates the burst of RNA synthesis. And then eventually this burst ends because there are diffusible histone deacetylases that recondense the gene. So even though you have not change the number of transcription factors in the nucleus, you have this on and off periods. And I'm going to leave you with this model, and just published in PNAS. You can go and look at it yourself. And there are many more controls we show there. And I'm going to show you the people who did the work. So this is Arjun who started all this. This picture is from a magazine that declared him as 10 most brilliant scientists in the country. And he started all this work here. And Diana Vargas, a few years ago, she made the first tandem repeats of these genes that allows her to see single molecules of RNA in the cell. Khyati Shah did her PhD here, and she was responsible for the work on C-Force and C-Jun and how noise propagates in the gene expression pathways. And Latasa Fraser, I'm sure most of you remember her. She carried out head tethering and noise in NYU. And Ryan Digdan is current MD PhD student. He did this Herculean task of sequencing human genome 16 times and did the ATAC-seq. And Salvatore Maris, there's these school probes that we have in this 17-year-old project. He made bulk of them. And here he just walks in with some beacons in his hand into my office. And I took a picture without telling him. And this is Fred, my guru and mentor. And he's the inspiration for all of us. I should say a very important comment about funding for this project. This is project going back 17 years, 18 years, and yet we never received NIH funding for this. Even though I had NIH grants during this period almost continuously, but they were for other projects. And this project, all three components of it were essentially supported by licensing income from molecular beacons and to a very small extent from Stellaris probes. So that goes out to say that, even though we work at the interface of industry and biology, we come back to real basic biology to do this. And thank you for coming to my seminar, and I appreciate your time. You can unmute yourself when you ask questions, I guess.
Jeff, can I ask a question?
Yeah, of course.
Sorry. Fascinating technique and talk. Thank you. So I'm just curious that we said that, again, that heterodimers, fast and genes, do you ever see them like on and off, or they just consistently? So like from the heterodimer, my main concern is that what is the negative regulation for these heterodimers? And my other question is that if you put the genes at a different distance from the centromere, do you see that differential regulate, like different, like do they regulate in the different mechanisms, or they have the same mechanism like you described?
So I should say that not all genes show great stochasticity. This is the work of the many other labs. They've shown that great majority of genes are highly expressed in a highly stochastic manner, and this is sort of the mechanism behind that. Some genes don't, like genes, things like actin and GAPDH, your classical housekeeping genes, they are constitutively on. And they are constitutively on because the system has, the polymerase doesn't have to find the gene. It turns out the polymerase is continuously located at that gene. And so that doesn't quite address the issue of subtelomeric region transcription. So I guess subtelomeric regions is some kind of heterochromatin, repressed chromatin. And so chromatin condensation status, repression status must play into this in a big way. But of course, we have not explored that. What was the question about C4 and C2?
I'm just curious at whether they form a heterodimer all the time, or there's one point that they fall apart. Whether there's what is the turnover?
Yeah, so basically, what happens is the heterodimer formation is also regulated by phosphorylation. So these transcription factors get phosphorylated by others, and that phosphorylation is necessary for them to form heterodimer and then migrate to the nucleus. And how their stability is controlled, I don't know.
So may I ask a quick question? So a much more fundamental, simple question. Very nice talk. I really enjoyed it a lot. And I love to think in stochastic terms, and we need to all do a lot of things in thinking about the probabilities instead of thinking everything is mass action. But my question is, we see a lot of studies of single cell PCR, we see RNA scope, and I wonder whether you would like to make a comment as to, how valuable and useful is that data, and how should it be analyzed? Because we see a lot of that stuff these days.
Yeah, so of course, single cell data, like the kind I showed you, you should not be looking at one or few cells. You need to look at many cells and then obtain an average. That average is going to have some meaningful insight, because ultimately, if this, if there's no serum induction, there will not be production of C4 and C2. So ultimately, the cell regulation is reflected in the amount of RNA, but you should look at average amount of RNA rather than from individual cell. So single cell seq is, of course, great commonly used technique, and people use that to explore both the heterogeneity and stochasticity, as well as so basically, again, you need to look at thousands of cells, and besides this stochastic gene on and off, you could have cells in different states of differentiation. So if you look at, for example, blood cells, and then look at doing single cell transcriptome analysis, most of the heterogeneity between cells or variation between cells comes from their being in completely different states of differentiation. B cells are different from T cells, and that drives their transcriptome. So within that transcriptome will be stochastic variation. So they are sort of overlaid on top of each other.
Right. So can I just add? So my question really was like perhaps taking towards the idea is an analysis of variance going to tell us something about specific genes in those massive RNA-seq data?
That's right. That's right. And also the variance is proportional to the expression levels. So it turns out that single cell seq just cannot see lowly expressed RNAs. And so the threshold of detection is something like 50 molecules per cell. And the most variation happens in the genes that are expressed, much less than that.
Sorry, I hand over.
Okay.
Very nice.
Dave, you're muted.
David.
Now I'm unmuted. Thanks, Jeannie. So that was really a lovely talk. So I guess in bacterial cells, the heterogeneity relies on usually positive feedback to amplify stochastic differences, right? So you have a poorly expressed gene, you have very few copies of a transcript, but some cells have a little bit more, but there's a positive feedback loop that amplifies that stochastic difference. And then I started looking at your model with chromatin compaction and so on. And it looked to me at first that there was no positive feedback component. But there really is, I guess. If I understood your model, that last slide, once you get some opening and some transcription, other factors intercede which amplify that initial stochastic event.
That's right.
Is that true? Is that
no,
the
it
way
is
you
true.
look at
In
it?
fact, the transcription cycle is meaning polymerase beginning the transcription, going to the end, releasing the RNA, and coming back. That is a cyclic event, and people have described biochemically for many years now that the polymerase that have just finished finds it easier to go back to the beginning of the same gene rather than going to a neighboring gene. And that happens for many reasons. One is that during transcription, there are many polymerases going through the gene, and they are polymerase-associated decompaction factors. So gene is sort of loose during this burst, and because of this recycling idea that there are transcription factors that remain bound to the beginning of the gene while the polymerase has left. So reinitiation is easier than fresh initiation. And I think that is important principle and is core of these bursts. I mean, of course, we didn't quite explore that here.
So the real stochastic event is the opening of the chromatin.
Exactly.
And this is a secondary event, which is like a positive feedback that amplifies that initial
event.
Stochastic event.
Yeah.
Yeah. Cool.
Thank you.
Sanjay? Yeah, no, I have a question.
Yeah, Jean.
Yeah, hi, that was really interesting. I loved it. So I have a question. Maybe you explained it, or maybe I just missed it. So with these stochastic events, if you take a cell and you keep looking at it over time, so presumably in that same cell, which was pretty low, later on, you're going to get a burst.
That's right.
Right?
Yeah.
So the ultimate final situation, even though you have stochastic events, since they go on and off and on and off, in terms of protein, which is much more stable, they're all going to be pretty much the same, no?
That's right. That's right. So even though, so it depends on what is the half-life of the protein. So those some proteins like C-Phos and C-Jun are intrinsically short half-life, and they will show a variation in the protein amounts, but others will not, or to less extent. So I think both kind of things will be seen depending upon the half-life and that's why the half-life of both RNA and the proteins and the complexes is relevant. I mean, this complex formation of proteins is very, very important to minimize the variation. So, like almost every protein works in complex. The ribosome have 50 proteins. So each protein is being expressed at some variability. So you make enough RNA, enough ribosomes forming complex, right? So how that plays out mathematically and so on has to be explored.
But just to continue, so since with time, the bursts will occur in all the cells.
Yeah.
Ultimately, they're pretty much the same.
That's right.
Yeah.
I tell you one more thing, Jeannie, it's a very important thing to consider. Arjun and several other labs have explored longer term stochasticity in the cell. So this, they've described a couple of hundred genes that are expressed stochastically over 5 generations, and then they go off. So you see same kind of bursty phenomenon, but the burst lasts for multiple generations of the cells. And that comes due to sort of compaction of the chromatin lasting multiple generations. And people are sort of saying that those kind of stochastic behavior allows cells to become drug resistant in cancer. And a very important, a very interesting new sort of different kind of stochasticity.
Yeah.
Thank you.
There are systems like that in bacteria where involving mutual repressors, where you can lock the cell into a new epigenetic state. So there's an initial stochastic event, and there are mutual repressors that can switch on and lock the cell into a gene expression state, which lasts for many generations. But it's not genetic. I mean, it's not encoded in the genes.
Yeah, it is
It
not
is genetic.
itself.
Right. Yeah.
Yeah, cool.
Yeah. Huh. Amazing.
Thank you. Anybody else? Good. So thank you very much for your time, guys. I appreciate it.
Thank you.
Thanks.
Great, thank you.
Thank you, David.